This video is going to be a review of information for our first exam for microbiology, and just a little information about how I plan to do this. Micro has a lot of information that is very straightforward, really obvious to get off the slides in the notes, and I have no intention of just reading off slides to you guys. So these videos aren't going to be completely comprehensive. My plan is to go through the more difficult and important conceptual stuff, and a lot of the very straightforward information that just requires some memorization, I'll let you handle on your own. And the first thing that I want to tackle is we spent a decent amount of time talking about setting up experiments and what makes a good experiment. And the beginning of that is testing a hypothesis. For the purposes of this class, our biggest restriction is feasibility. Not a whole lot of money, not a whole lot of materials. And oftentimes the simplest experiments are the best, though. And so the first thing to think about is what can you test? Are you able to use human subjects? Are you stuck using just bacteria in a lab? Could it be done on animals? What are your options? The second part of that is, once you do have an experiment set up, you have to make sure that you're setting it up in a way that you're actually going to get an answer that you can interpret. The first thing to consider with that is you only test one variable at a time. The easiest example of this is, we did a lab in class where you tested the effect of either hand sanitizer or hand washing. If you take hand washing as an example, the thing that you really want to test is, does hand washing have a strong effect compared to not washing your hands at all? Or perhaps you could test, how long do you need to wash your hands, or what kind of soap do you need to use? But you can only test one of those variables. If you're testing time, 15 seconds versus 30 seconds versus a minute, that's fine, but then the kind of soap and the temperature of the water and the way that you rub your hands together, all of those things should be held completely constant. If you start manipulating more than one variable at a time, there is no way that you can know which one of those variables actually caused the effect. And the trap that a lot of people fall into, especially with fairly simple experiments like that, is if you start trying to mess with more than one variable at a time, you can't really interpret anything because everything comes out confusing at the end. The second really important consideration is positive and negative controls. The most important one by far is the negative control, which you always have to have. And a negative control is really just a condition that you expect to not work, to not give you a result. And so, for example, in a condition like if you're testing hand sanitizer or the effect, the effective ingredient in hand sanitizer is alcohol. And so if you wanted to see, a negative control for hand sanitizer, you could get a liquid that is a sort of a placebo and feels like hand sanitizer but doesn't actually have any alcohol in it. And you expect it to not work because you just took out that active ingredient that's actually killing bacteria. That would show you no result. The bacteria should all still be there. It shouldn't have an effect. Right? That lets you know that there's not something strange happening in the experiment. A positive control is something that works very well. And an example of that would be if you're testing hand sanitizer, a positive control might be diluted bleach, for example, or another really powerful disinfectant. You expect it to give you a really strong result. This is just another way of ensuring that your experiment's working correctly and you can actually see the results that you're expecting. In clinical trials involving medication, negative controls are almost always placebo, a pill that just doesn't have anything active in it to make sure that it's not a psychological effect on the patient. And positive controls, if you have one, would just be another drug that you already know works. So if you're testing, a new painkiller, perhaps to treat headaches or muscle soreness, you could use ibuprofen or acetaminophen or something like that as a positive control to make sure that you can even measure an effect. The last thing is you can't just do an experiment once. In order to be accepted by other people, an experiment needs to be repeated multiple times, Ideally, it needs to be done by different people as well, just to make sure that one person doesn't have a bias or they're doing something incorrectly. That just makes you more confident that the results are real and not just a fluke. Right. The last step in all this is, after the experiment's over, you're going to have to interpret the results, interpret the data somehow. And the biggest thing to decide is, and this sounds strange when you first hear it, but you have to decide, are you looking for a difference or an amount of difference? And what I mean by that is, if you have a very, very small difference, coin flips is the example that I use very often because it's easy to imagine. You can imagine a coin that's rigged so that on average it comes out as 51% heads and 49% tails. Okay. If you bet money on that coin and you kept betting on heads, it would take a long time, but eventually you would start winning a lot of money because on average it will come out more often in your favor in the long run. Now, it's not that big of a difference. So in order to detect that, you would have to have a really, really, really big sample size. Okay, flipping this coin 100 times isn't going to really make you confident that it's 51-49. Flipping the coin 1,000,000 times would make you really, really confident, because it should come somewhere around 501,000 heads and 499,000 tails, which at that big of a sample size is a significant difference. All right. If this was a drug, let's say that instead of being heads and tails, you could imagine that the placebo, right, helped 49% of patients, and the actual drug helped 50% of patients, 1% difference. That's really not all that important. If you had a very, very big sample size, you could detect that difference and get a good p-value. Okay. Remember, the p-value is it's a statistical calculation that tells you what's the likelihood that my results are due to chance, that they're just a fluke. And if your difference is small, having a really, really large sample size, in statistics that's called the N number. So having a large N, large sample size, can allow you to detect this small difference, but that doesn't make it important. In a clinical setting, you really want to know what is the amount of difference, and that's a little bit more difficult. For amount of difference, you can measure it two different ways. One way is relative risk or relative effect, and the other is absolute. As an example of that, let's say that in your trial, your placebo had a positive effect on 8% of people that took it. And let's say that your drug had a positive effect on 10% of people that took it. Okay. Now, you can measure this relatively or absolutely. On one hand, you could say that 10% is an increase of 25%. Right? 10 is 25% more than 8. And so relatively, you got a 25% bump. Okay? But in absolute standards, you got 2 percentage points. Right? 10% is 2 percentage points more than the placebo. They're both technically true, they're accurate numbers, but the problem with the relative number is it doesn't really give you a whole lot of context. And the thing that's been done, starting to be done more recently, is you can convert this absolute number into something called an NNT, which we talked about in class, the number needed to treat. And if you imagine that this was given to 100 people, right? If 8% received a benefit, that means that there were 92% up here that didn't, and if 10% received a benefit, that means that there were 90 up here that didn't. Thinking about it that way, giving the drug helped 2 people, right? These 2 that didn't get a benefit in the placebo moved down to the benefit category here. They got a benefit. So you helped 2 out of 100. All right, so the NNT is 100 divided by 2 equals 50. Another way of phrasing that is you had to give this drug to 50 people before you helped one person. Right? That sounds very, very different than someone saying 25%, right, increase in benefit. So they're all true numbers, but they can really skew the way that somebody interprets the data if they don't know exactly what they're looking at. Okay, next topic to talk about is I spent some time going through details of bacterial cell structure, a little bit on viruses, a little bit on eukaryotes. I want to try to tie all that together. This is, a very generalized mock-up of a prokaryotic cell. For us, that pretty much means bacteria. Archaea are also prokaryotes, but we don't really care about them from a disease standpoint. And what I want you to do is not memorize all the little parts of a bacterial cell. We're going to look at some because they're important. But what I want you to think about is when you're treating infectious disease and you're using tools like antibiotics and your immune system is using things like antibodies, the big thing that you're after is you want to find something in this bacterial cell that is unique and different from humans so you can target it. Easy examples of that. Cell walls are very important to bacteria. I'll get to that in a little bit in more detail. That's really significant from a clinical standpoint because if you can interfere with or mess up or damage a bacterial cell wall, you can kill the bacterium. And that's a nice target because humans don't have cell walls, and so they're very easy to go after. Right? On the other hand, bacterial DNA, like its plasmids, chromosomes, the way that DNA is copied and replicated in bacteria is actually really similar to the way that it happens in humans. Not really a very good target. And so when you're looking at prokaryote, bacterial anatomy, the big question from a medical standpoint is, what parts of this bacteria can we tackle, can we try to interfere with for drug therapies? And zooming in a little bit closer, I just want to remind you, cell membranes in bacteria look different in a lot of ways compared to eukaryotic cell membranes, but the general principle is the same. Right? This would be intracellular, this would be extracellular, and the thing I want you to focus on, because this comes into play later, is in a living bacterial cell, right, whatever is on the outside of the membrane, like these big purple blobs are all proteins embedded in cell membrane. This is all that your immune system can see. Right? Anything that's intracellular inside here is hidden away. You're not going to get inside of that until the cell dies anyway. And so these targets are very, very important. This is what your antibodies stick to. A lot of these surface proteins are what vaccines are based off of because it gives your immune system a shot at seeing what the bacteria or the virus looks like. And so membranes and their surface proteins are really important for recognition purposes by your immune system. Okay, I mentioned cell walls earlier, and we've done gram-positive, gram-negative staining in the lab, so you can sort of visualize the difference between those. And the gram-positive, gram-negative designation is really just based on differences in cell wall structure. Right? Cell wall is a rigid layer of protein called peptidoglycan. Right? Same molecule in gram-positives and gram-negatives, just it's much thicker in positives. And the whole purpose of that rigid layer, peptidoglycan, peptido for protein, glycan for sugar, for carbohydrate, it's just a big mashup of protein and carbohydrate that makes a thick, dense wall whose main purpose is to resist changes in pressure. Remember, bacteria aren't like animals and humans. They don't have kidneys that can keep osmotic pressure stable inside their cells. And so if they get thrown into a very watery solution or a very high salt solution, they're going to face osmotic pressure and potentially rupture or shrink. And that rigid cell wall is just a barrier that prevents that pressure from actually causing an effect. Also, like I mentioned before, because cell walls and peptidoglycan are so unique to bacteria, they make really good drug targets. And the very first antibiotics ever developed, antibiotics based off of penicillin, they did exactly that. They target cell wall synthesis. And as long as there's not resistance, that works very well. Okay, so the anatomy of it, gram-positive cells, straightforward. Cell membrane on the interior, surrounded by a very thick peptidoglycan cell wall on the exterior. You can see the membrane colored yellow and the peptidoglycan layer colored brown here. This explains why gram-positive cells show up very dark purple under the microscope after the gram stain. That's because that purple dye that you use gets taken up by that peptidoglycan layer. It's very thick, and so it retains a lot of the dye. Gram-negative cells have this very thin peptidoglycan layer. Same molecule, same purpose, but since it's so thin, it doesn't really take up much of the dye, and that's why gram-negative cells have that faded reddish-pink color under the microscope. Other main difference, gram-negative cells have a second outer membrane outside of their cell wall, and so the cell wall is really sort of sandwiched between two membranes. And that outer membrane will come into play later. We'll talk about it. When gram-negative cells die, that outer membrane can become kind of a problem and irritate your immune system. Okay, next topic to tackle is bacterial, at least a brief intro into bacterial DNA and genetics, some unique parts of it. The first thing is bacteria have chromosomes just like human cells do, except they only have one, not many like animal cells do, and it's circular. And the circular part of that doesn't really mean a whole lot to you. It sort of changes the way that they replicate, but it's not that big of a deal. The important thing to remember is a bacterial chromosome is just a big piece of DNA, the same way that your chromosomes are just big pieces of DNA. And they code for proteins the same way that the genes on your chromosomes code for proteins. Now, they are haploid, which means they only have one copy of that chromosome, not two copies the way that the humans do. And everything inside of a bacterial chromosome, that's everything that's really necessary for survival. They've got all that they need there. That's all their essential DNA. And you can see here these little yellow coiled masses. Those are circular strands of bacterial chromosome DNA that are all coiled up on top of each other. Same thing that's being illustrated here. Now, the bacterial chromosome has all of the essential DNA that's necessary for that bacterium to survive. But bacteria have evolved this very it's beneficial to them, it's very bad for us, this beneficial trick where they can use other pieces of DNA, smaller pieces of DNA that still contain genes, they still code for proteins, but they're not really essential. And we call those secondary pieces of DNA plasmids. And so a plasmid is just a small piece of DNA that codes for one or more genes. It's not absolutely essential, but if that plasmid has a gene that gives the bacterium some kind of benefit, then it's going to be maintained. They're going to keep it. Examples of this, genes on plasmids can code for enzymes that degrade antibiotics. This is the most common way that bacteria develop antibiotic resistance. If you treat a patient with an antibiotic, like penicillin, for example, but this bacterium has a plasmid that lets it produce an enzyme that can destroy penicillin, it's now resistant. The drug's not going to have an effect because the bacterium can just metabolize it away. And not every plasmid will have those genes, but some do. It's also possible that plasmids could carry genes that code for toxins. Almost every toxic molecule that bacteria produce, the ones that really harm you, they're proteins, which just means that they're genetically coded for by genes, sometimes on plasmids, sometimes in chromosomes. But the thing that makes plasmids sort of extra dangerous is that they can be transferred, and that's what this is showing over here. So it's not like a chromosomal piece of DNA where it can only be passed off to offspring. Plasmids can actually be copied, passed on to a completely different cell, and it's rare, but it is possible that this new this recipient cell, the one that got the plasmid, could even be a different species or strain of bacteria. So what that means is one bacterial cell develops resistance. It's possible for it to take a copy of the piece of DNA that codes for that resistance enzyme, pass it on to another cell. This increases the rate at which resistance can spread and makes it really difficult to keep control of. Right, so that's how plasmids can be really quite dangerous. The one upside to them is they make a really important tool in a lab. And the thing that plasmids allow, a bacterial geneticist or a molecular biologist to do is it's now possible for a scientist to make their own plasmid. This is an example of a map. This circle is supposed to represent a circular piece of plasmid DNA. And the thing that you can do is you can take that plasmid and splice in or insert any gene that you want. And so an example of how this would work is, let's say that I want to mass produce a particular protein. This protein might be part of a vaccine. It might be used for some sort of commercial application. Lots of different possibilities. If you put that gene inside the plasmid, I'll just call it gene X, whatever you're interested in, and then you also put an antibiotic resistance gene. This AMP-R stands for ampicillin resistance. Ampicillin is a common antibiotic used in lab settings. What you can do then is add this plasmid to a bacterial culture, and a few of those bacterial cells will take up the plasmid. Right? They'll take it up from their environment. And what you can do then is, if you grow those bacteria on a medium that has the antibiotic, like ampicillin, only those that have the plasmid can survive, which means they now have your gene X, in them. And so if you keep growing those bacteria with the antibiotic that you've given them resistance to, you guarantee that they keep producing the protein from the gene that you're interested in. Right? You now basically have a little protein factory that you can use those bacteria for to make whatever protein you happen to be interested in. Okay, next consideration, bacterial endospores. This is usually not a big concern because most bacteria do not form endospores, but some do. And several of them are fairly important bacteria, like anthrax, for example, is spore forming. Most common in bacteria that live in soil because they often have to face harsh conditions. And all the spore is, it's really just a dormant, you can sort of compare it to hibernation, only it's a very extreme form of that. And it's a way for bacteria to survive really difficult environmental stresses. Normal bacteria that can't form spores, if you leave them out on a dry surface for more than a few days, they'll die because they just dry out and fall apart. If you can form a spore, what that essentially allows a bacterium to do is form this very dense, dehydrated capsule where all the water gets drawn out of the cell. You compact the DNA into this tight little ball and just hang around and wait until conditions get better. This is meant to help these bacteria withstand really difficult conditions like heat and drying or desiccation, radiation, chemical poisoning. The problem is all of these things, heating, drying, freezing, etc., these are the primary ways that we try to get rid of bacteria. And so if you're trying to get rid of a spore-forming bacterium, you have to go to pretty excessive lengths, or extensive lengths, sorry. The best example of this is if you work in a lab or a clinical setting where things need to be disinfected, completely sterilized, you'll often use something that looks like a giant pressure cooker called an autoclave. And an autoclave just heats to such enormous temperatures, way past boiling temperature, that even spores can't handle it. And so they can be kind of difficult, but there are ways to get around it. Okay. Last thing, bacterial shapes. This is pretty straightforward, so I'm just going to let you see the pictures. Just remember, in lab, everything that we see are either going to be spheres, cocci, or rods, also known as bacilli. These other shapes you'll see from time to time, but probably 95% at least of the bacteria that infect people fall into one of those two categories. All right. Last thing in the prokaryotic section, I mentioned archaea. Just remember, archaea are prokaryotes. They're found in a lot of extreme environments, but they're not found in humans. They're really non-pathogenic, and so we don't really care about them here.